So we're going to do another example of this kind of identifying a specific organism and then looking at a microbial population. And the paper we're talking about is this paper from Applied Environmental Microbiology. It's a little old now, but it's got some really cool stuff in it. Novel forms of structural integration between microbes and a hydrothermal vent gastropod from the Indian Ocean. Now, what in the world does that mean? Well, they've got the snail. So they go down in these deep sea submersibles to hydrothermal vent fields. And so this particular one, this is a picture, I think this is from Guaymas Basin off the coast of Washington. But it's the same sort of thing. You've got a spreading center where two plates of the crust are opening up or compressing together. Usually it's opening up. And the water from the ocean nearby seeps down into the crust and then gets heated, just like in Yellowstone, and comes up to the surface. When it comes up to the surface, this water, the pressures, this particular site's 1,000 feet underwater. The pressure means that the water can be really, really hot. Some of these places have water that comes up out of these vents at 350 or 400 degrees centigrade as liquid water. Obviously, that mixed instantly with 2 degree or roughly 2 degree surrounding seawater, and there's a lot of chemical potential there. The hydrothermal vent fluid is at equilibrium in its environment, in the crust, very high temperatures, lots of hydrogen, lots of minerals dissolved in it, but it's at more or less equilibrium, chemical equilibrium. And the surrounding ocean water is more or less at chemical equilibrium. But when the two mix, that mixture is well away from chemical equilibrium. There's lots of reducing power, there's oxygen from the seawater, and so there's potential there for organisms to make a living, and they do. Most of it is by sulfur oxidation. And so you have reduced sulfur compounds and oxygen mixing together, and they can make a living. And these things then form kind of an oasis on the seafloor. You have microbes growing on it, and then you have animals feeding on the microbes, and it goes up the food chain. And so here's a field of the tube worm Riftia, and some tiny shrimps living around in here. There's some clams. A really interesting thing happens, though, which is some interesting symbioses between these kind of animals and microbes. And so this organism, Riftia, it's a tube worm. You see these in aquarium shops. They're called feather dusters. And when you startle them, they suck down into the tube. This animal doesn't eat. Doesn't have a digestive tract. It is not a filter feeder like most tube worms. Instead, these gills, which is what they are, absorb chemicals from their environment. And in specific, CO2, some nitrogen, and sulfide. Now, sulfide is pretty toxic to animals, right? Why is that? Why is sulfide toxic? It's in hemoglobin, right? It binds hemoglobin very tightly. And so these organisms have a specialized form of hemoglobin. That oxygen, the CO2, and sulfides are carried by the circulation into a specialized organ within this animal. They have no digestive tract. And in that organ, there are bacterial symbionts, sulfur oxidizing symbionts that carry out that reaction and feed the animal in return. These things get to be like 6 feet long. They're amazing, like this big around. They're really big. There are clams that do this too, and they have symbionts in their gills. There are shrimp. These shrimp are decorated with little spines and things all over them. And sulfur oxidizers grow on that hard surface, and they sit there and preen themselves, feeding on that biofilm all day long. So the symbiosis in there are really, really interesting. The site we're looking at today is a place where two plate spreading centers come together in the Indian Ocean. This place is 2,400 meters deep. That's a long ways down. And what they found was a snail. that they'd never seen before. Here's a picture of it. And they knew right away that this was a very special animal because it's covered in scales. It lacks one thing that every other snail species has, which is an operculum. You guys know what this is? Yeah, so here's a queen conch. A little worse for wear. These animals are bivalves. They don't look like clams, but they're bivalves all the same. This is one shell, and the other one, which is completely flat, folds over. When the animal retracts into the shell, it covers the animal. It fits into that opening and protects the animal from predation. That second shell, the operculum, can also be used for a variety of other purposes. Cone shells, little pyramidal-shaped things, have an operculum that's very thin, very fine, and contains a poison. And it's used essentially as a spear. They spear fish and eat them. This particular animal, the queen conch, uses the operculum to dig in the sand, and so it buries itself in the sand and moves around collecting food. Even land snails have an operculum. That oval thing that sits at the back of the animal as it trails away is the operculum. This one doesn't have an operculum. But it does have scales. And so it can't retract into a shell, but it does this instead. It folds its foot in and presents to any predator this set of scales like so. These scales are armor plated. They're covered in a layer of greigite and iron sulfide. So there really is metal plating on the surface of these scales. They're very tough. The black here, by the way, is not greigite or any iron sulfide. It's just a standard pigment that the organisms have. So that's a different color. So when you dissect this animal, you find something really interesting, which is that it does have a digestive tract, but it's clearly non-functional. So these snails have a radulus, it's a tongue with an abrasive surface that they use generally to scrape surfaces for algae and biofilm and that sort of thing. The radulus on this animal can't even be extruded from the mouth. So it can't feed. The digestive tract is open, but when they dissected animals, the only thing they find in there are bits of iron sulfide and that sort of thing. There's one bit of the digestive tract, though, which is not rudimentary. And that's the esophageal glands. You guys know what esophageal glands are? Right up at the end. They're used to lubricate your esophagus, right? When you swallow something, that stuff doesn't slide down on its own like magic. There has to be a lubricant there to allow that to happen so that the peristalsis in your esophagus can push it down. The esophageal glands provide that lubricant. And so here's a cross-section of one of these animals. Here's the foot and the scales. The gills are up here, which are usually in the shell. And this is the esophageal gland. They're huge. And when you take a cross-section through the esophageal gland under an electron microscope, what you see is that the cells of the esophageal gland, here's one cell, here's the nucleus, are full of bacteria. This is what they look like in cross-section. Now, you can't keep these snails alive. When you bring them up to the surface, they're dead. And so they're a little hard to study that way. So there are two questions that these folks had about this animal. Number one, what are these bacterial symbionts providing to the animal? The hypothesis would be that, like in Riftia and other deep-sea vent animals, some of them, that it's providing carbon to the animal, right? That sulfur and oxygen go in, and some kind of fixed carbon comes out. The other question, and so they want to ask that, who are these organisms and what are they providing to the animal? The second question is, how do these scales get their armor plated? Because no other mollusk does any such thing. So what's going on there? So the first thing then was to dissect some of these animals, pull out the esophageal gland, and do ribosomal PCR, right? Standard issue stuff. And maybe to pleasant surprise, they get one sequence, and one sequence only. That organism trees out right here amongst the gamma proteobacteria, amongst a bunch of other thiotropic creatures, so sulfur oxidizing gamma proteobacteria, including some symbionts of other deep sea vent animals. Also some free-living sulfur oxidizers in this environment. Raises an interesting question. How do these animals get those symbionts in the first place? Are they maternally inherited somehow? Or do they pick them up from the environment as larvae before they mature? Probably the latter in this case. Although it's only a single species in all it's the same species of thiotrope in all the snails that they dissected. Here's a close-up of that. And so here's the scaly snail endosymbiont, and it's closely related to this free-living vent thiotrope, and pretty closely related to the symbiont of Riftia, that giant tube worm I showed you.
So that's one species?
One species. And so either when the females lay eggs, they plant those organisms there so that the hatching larvae get them, or they're floating around in the environment and there's some kind of host symbiont recognition that allows that specific symbiosis to take place. That wouldn't be so unusual. The squid, I can't remember which squid it is, but it's got a light organ that it glows in deep sea. Excuse me? Yeah, it was Vibrio Fisher. Cuttlefish that's a different thing in cuttlefish. That's the actual animal cells that can do that. But in the case of this squid, it's the bob-headed squid or something like that. There's a specific strain of the Vibrio fisheri that it associates with. Vibrio lives in the ocean anyway, but it can form a specific symbiosis, and it may be something like that here. Or it may be that they're planted in the egg somehow. We don't know the answer to that. Or at least I don't know the answer to that. And so the question then is, okay, we can't grow these snails, so how do we figure out what the bacterial symbiont is providing? It's not like we can go down there in a submersible and feed these snails radioactive sulfur or something and see what happens. But you can look at stable isotopes. And so you guys know, I'm sure, that enzymes typically are rate limited or at least limited to some extent by diffusion. And that light isotopes, because they diffuse faster, they're lighter weight, right? They diffuse faster at any constant temperature. Enzymes tend to process lighter isotopes of substrates faster than heavier isotopes. And so products are usually enriched in lighter isotopes. And how much that enrichment is depends on the enzyme. Some enzymes have a greater enrichment than others. This is one of the ways you use to see how an organism is fixing carbon. And then as that lighter product moves through the food chain, it gets homogenized because it also is then processed faster with lighter isotopes. As it moves up the food chain, they become less and less enriched for lighter isotopes. And so they decided then to test tissue samples of this animal and other animals in the same environment for light carbon in their tissues. So that's what this was, delta C13. The C13 is a heavy isotope, and so they're looking for a depletion of heavy carbon in their tissues. And the closer to that carbon fixation it is, the more, or the more that depletion will be. The greater this number will be negative. And so what they see is in the scaly snail, the esophageal gland tissue has a depletion of 20%. Yeah, 20%, which is easily measured. If you look at other tissues of the same animal, it's actually a little bit less depleted, as if that carbon has already gone through some processing after being fixed. Remember that the esophageal gland carbon will be a mixture of bacterial and host tissue. If you look then at other creatures, these things are grazers. And so this thing is a grazer, and this thing is a predator, and their depletion is much less. That is, they're already starting to process out that light carbon. And so the implication is this thing probably, the esophageal gland probably really is where carbon fixation is taking place. This raises a really interesting question. What about nitrogen? If you absorb sulfur and CO2 from the environment, oxygen, and fix carbon, the next limiting thing is going to be nitrogen. If you're an autotrophic snail, which is what these are, right? You've got to have a source of nitrogen. And if you can't eat, where do you get it? Well, they see the same isotope effect in reverse. These are positive deltas here. They see the same isotope effect for nitrogen as well. So not only are these endosymbionts providing fixed carbon, they're providing fixed nitrogen as well. The next limiting factor is phosphorus, and that just has to come from the environment because there's nowhere else to get it. Yeah.
What is the form of nitrogen that's fixed as opposed to not?
So generally, these are going to be homogenized tissue samples, so that's going to be generally amino acids. And presumably that comes from dinitrogen gas in the environment, which is absorbed and then fixed by the organism in the tissue. As an aside, this has been confirmed since the genome sequence of these organisms has been done, and their nitrogen is it really is a nitrogen fixation. And they're fixing CO2 by the counting cycle. Well, that's pretty cool. You know how the snail makes a living now, right? It floats around, it doesn't float around, it crawls around the surface, and it's absorbing gases from its environment through its foot. And the venation of the foot is hugely abundant. So they're exposing their circulatory system on this foot for the ready exchange of gas. Not through the gills, which is where you would usually expect it to be, but through the foot. And so the blood flow is from the foot directly to the esophageal gland and then off to the gills from there. So they're absorbing things through the foot and then blowing off waste products into the gills. Well, that's really cool. An autotrophic snail, an autotrophic nitrogen fixing snail. What about that armor plating? That's a little more complicated. So this is a cross-section of the animal. Here's the base of the foot, and here are some scales as you're slicing through them. And this is what one looks like picked off. This is a series of EMs that have been put together again that show you the structure of one of these scales poked right through the middle. So this is one surface through the scale to the other surface. And the dark stuff you see there is all metal. Metal sulfides and greigite and that kind of stuff, irons. And you can see that it's very highly enriched on the surface here. The scale is otherwise made of just exactly the kind of stuff you would think it's made of. Concholine, the protein that these shells are made of before they're calcified, that kind of stuff. And when they look at the surface of that, so if you take this and zoom in, here's the surface of the scale. This is all iron sulfide. But look at all this stuff on the surface. This is what it looks like when you look a little closer. This is a biofilm. So these scales have a very specific heavy coating of biofilm. Now we're pretty sure that snails don't know how to make armor plate. Maybe these do. The hypothesis would be that it's this biofilm that's producing the iron sulfide plate, not the animal itself. So, who's in there? Yes. Yes, so it's clearly a specific symbiont of the organism in the esophageal gland. But it also has a symbiosis with the surface. And that's not, I mean, we all have biofilms on us, right? And microbiomes in various parts of our body. That's not unusual. The unusual thing would be what it's doing, if that's the case. So, and I'll cut to the chase. They still don't know if it's the biofilm that's producing that plate. And so they did ribosomal analysis of DNA extracted from the biofilm on the scales and the biofilm on the surface of the shell for comparison. Because the shell doesn't have that armor plate. And so they got a bunch of sequences. These are just the ones from the green non-sulfur bacteria and cytophagas. They're all over the place. There's a bunch of them. Here are the rest of them. Primarily epsilon proteobacteria, also some delta proteobacteria. Turns out epsilon proteobacteria, I mean, we talked about epsilons a little while ago. The ones you're familiar with are things like Campylobacter and Helicobacter, gut symbionts. It turns out, though, that the sulfur metabolizing epsilon proteobacteria make up a large fraction of the kind of organisms you find on the seafloor. Whether that be in a hydrothermal vent environment, a traditional, ocean bottom ooze environment, whale drops. So when a whale dies, it sinks to the bottom and creates this giant oasis of sulfur and nitrogen and stuff. Epsilon proteobacteria are probably the predominant bacteria in deep ocean floor environments. And so having them here is no particular surprise. I think you can see one thing really easily, though, because in this case, the ones I've highlighted green, are associated with the scale biofilm, and the ones in blue are those associated with the shell biofilm. The shell biofilm is obviously a much less complex community by quite a lot. And so you've got, some gamma proteobacteria, a bunch of epsilons, and that's about it for the shell. There's no evidence that anything unusual is happening on those shells. And this is exactly if you look at this population and look at its diversity and the community structure, it's the same as any other solid surface in this environment. These are just organized organisms that are colonizing the solid surface more or less non-specifically. There's no evidence that there's anything special about the shell biofilm. The scale biofilm is much different. It's clearly very highly diverse. Here's some other ones, all these other biophils. And I showed you the cytophagas and green non-sulfur bacteria. These guys are probably the filaments that you're seeing in the biofilm. And so you've got a very, very rich, very diverse biofilm on these scales, not what you expect for just a solid surface. And so they conclude tentatively that it's probably that biofilm that's producing the iron sulfide plating on the scales. They haven't done an experiment to prove that, though. That's a tough one. How would you do that in the absence of the ability to grow the animal in the lab, or go down there and spend hours or days with the animals in situ? Yeah.
Do you think some sort of model like create an iron sulfur or like a shell type coating and inoculate and see what happens?
Yeah, so my guess is there are folks doing those kind of experiments. but I haven't seen any results from that. An interesting thing about this is that the biofilm is here, but the iron sulfide is not just on the surface, but it's also within the interior of the scale. And so on one hand, you might say, well, it can't be the biofilm that's producing the iron, but you have to remember that these scales don't appear from nowhere. They start out on just as the surface of the foot, and then grow from there. And so it may be that this was the surface at some time previously, and that the scales grow out from that, kind of like the way a tree does, right? And so, and that absolutely is the case for shells on a snail and the operculum, and so presumably these things as well. So these things would have rings on them that represent the, I don't know if you can see them on this, but there are rings on here that show its progressive growth. And so these then may have represented the remnant of that iron sulfide plating in the past in the animal. There's one experiment they didn't do here. Can you guys think of what that might be? Think specifically about the esophageal glands. You're trying.
Well, it seems like they only got one sequence, that were not sequenced.
Wouldn't it be nice to do a fish experiment and prove that that one sequence you've got really lights up all the bacteria in those tissues? Not just that they light up some of them, but they light up all of them. That experiment was not done. Instead, what these folks did much later was to extract DNA and sequence the genome of the organism. And it turns out to be all the things you would expect of it. It is the organism that they got ribosomal data from, and it's got all the properties you would think. In fact, it looks a lot like the generic Cola, except that it's an iron oxidizer. Excuse me, a sulfur oxidizer. One thing I want to talk with you about, though, it has to do with the process of collecting all these sequences. When you do this experiment, you've got a mixture of DNA from all kinds of organisms. And you do a ribosomal PCR. There's a huge artifact that can show up, and it does show up in almost every experiment that's like this. And one of the things you have to do is eliminate these artifactual sequences. These things are called chimeras, and when you look at the paper, you'll see they talk about chimeras. So what is a chimera? Chimera is an ancient Greek creature that was supposed to be a hybrid between a lion, a snake, and an antelope. Think jackalope. You guys all know what jackalopes are, right? So what's a sequence chimera? If you're doing a PCR reaction, you're amplifying some piece of gene. The reality is these enzymes aren't perfect. And it happens all the time that the polymerase will stall, particularly in ribosomal sequences where the single-stranded DNA wants to fold up into the same kind of structure that the RNA that it encodes will fold up into, right? Lots of stem loops and all that kind of stuff. And the DNA polymerase will hit one of those regions and pause. Most of the time, it will then bind some more nucleotides and roar through from there, unzipping any structure in the process. But sometimes, either it runs out of time, or it simply falls off the template or something like that. And you end up then with partial DNA fragments. When those DNA fragments end in sequences that are highly conserved, they can serve as a primer on a different sequence. They're complementary because it's a highly conserved sequence. It will bind, and then in the next round of PCR, another DNA polymerase molecule will bind this like it does every other primer template complex, and make the rest of the molecule. Now you've got a sequence that's part from one organism and part from another. And so when you get your sequence collection, most of the sequences are fine, but occasionally you get some that are like this. And you have to have a mechanism to recognize those. One of those mechanisms is to take the RNA that this would encode, fold it up into a secondary structure, and if distant parts of it can base pair properly, then you believe that it's real. There are other mechanisms. Check chimera is a good one, where it identifies, it takes each end and identifies the closest relative in a database of that short sequence. And then you just plot the relative similarity of the sequence that this matches to the sequence that this matches as you scan down the sequence, and you end up with a plot that looks something like this, where this is the junction point. And any of those sequences you throw away. Now, it's really easy to find one of these chimeras if these two sequences are very, very different. If one of them is a yeast and the other one is an E. coli, there's no mistake in the fact that this is a weird RNA and must be a chimera. But what if one of them is E. coli and the other one is salmonella? You're never going to detect the fact that it's a chimera. You can't do it. Does it matter? Because your ribosomal analysis can't resolve those organisms anyway, can it? So these chimera functions, they're not perfect, but whenever you do an environmental population analysis like this, you've got to do some quality control. Chimera checking is one of them. High throughput sequencing, there are mechanisms in the barcoding process, which we'll talk about later, that are used to check the quality of the sequence so that you can go through your database and throw away anything that's questionable. So have a look at this paper. It's really interesting. And take a look at what's come up since then on this animal. Because there's some folks here and nearby that are working on it. Yeah. So as a plant, is this tree over here, is that an autotroph? Yeah, so I think you're in one of these gray areas here. Because this plant, the actual eukaryote of that plant is not autotrophic. It's the chloroplast, the symbiont, that does that. That's a little bit dangerous argument to make, I'll agree, absolutely. Because by that same criteria, you're not an aerobe. And so that distinction between what's something else, what's another organism, and what's part of you, I think there's a giant gray area in these kind of symbiosis. This snail cannot survive without the bacteria. It's like an aphid, right? If you treat an aphid with ampicillin, it dies. Yeah.
Can the bacteria survive without snail?
So that's an interesting question. It's never been found just in the water generally, but it must be there. I'm not aware that anyone has ever cultured that way. My guess is it's alive. So we were just talking with the guy who teaches chemistry here before. There are organisms that are adapted to live at high pressure, but it turns out that pressure doesn't have a lot of impact on creatures unless there's gas as part of the body. Gas is compressed, right? Water has almost no compressibility. Living at high pressure is the major effect on that. If you don't have gas bases, well, solubility of gases changes dramatically. That's not really an issue for these things. But it's lipids. Lipids are more compressible than water, and so this has an impact on membrane thickness. The more pressure it's under, the thinner your membrane gets. And that has an effect on the ability of that thing to act as a capacitor and that sort of thing. So organisms at these pressures have altered lipids. But in general, organisms can be pulled up from the deep sea and they look just fine. delicate, so you have to treat them that way because they're in, the environment's very stable and motionless. They also be sensitive to light and dissolved oxygen is a big one. So complex animals don't do very well when you dig them up in the ocean, but bacteria, you can grow all kinds of stuff. Yeah.
With that process, these growing creatures that can be depending on how you walk about.
Yeah.
I usually have like some sort of pressurized environment.
So you can do that. It actually turns out to be remarkably simple to keep an environment like this under pressure. If you've got a stainless steel container and you screw a lid down very tight on it, if that container won't expand when you bring it up, it can still be under tremendous pressure when you bring it up. It's all liquid pressure, so it's not as dangerous as a compressed gas. The biggest issue with bringing these kind of creatures up is you have to do it slowly to let any dissolved gases evaporate from the organism in advance. In a human, this is called decompression sickness, right? But now a diver goes 100 feet underwater, not a mile and a half. So that's a big issue. Not so much oxygen, because there isn't much dissolved oxygen down here, but lots of nitrogen. And so these creatures will get decompression bubbles in them and it tears them apart. Not so much bacteria, obviously, which are on a different scale. All right, so next time, remind me. Oh, we talk about the human microbiome. Really cool paper. Very interesting beginning survey of the human microbiome. Where is it? Here we go. This is a Rob Knight paper from the University. At the time he was in Colorado, Boulder, Colorado. It's taking samples from all over the body from a bunch of people at different times and looking to see who's there. And some real surprises. All right, I'll see you guys next time. We're done a little early.